Hello, everyone. I'm Beth Johnston. I'm the Chief Community Engagement Officer here at the FSHD Society. And together here with my colleagues, June Kinoshita and Dr. Jamshid Arjamand, we welcome you to today's FSHD University webinar. As you know, the FSHD Society offers these free webinars and so many other educational events to educate you and empower you and everyone affected by FSHD to really become their own best advocates. So today, with much anticipation, we welcome Casey Childers, the Chief Medical Advisor, and Dr. Alexandra Collin, Head of Therapeutics from Epic Bio. It's a San Francisco-based therapeutics company who's actually developing a gene therapy medicine specifically for FSHD. So without further ado, Dr. Collin, if you'd like to take it away.
Absolutely. Thank you. Thank you very much for having me. And, we're very excited to talk to everybody today. So I'll start with sharing my screen and share a little bit of a presentation today. Here we go. Can you see my screen?
Yes, we're seeing, yeah.
Okay.
There we go. That's better.
All right, so thank you again. And I know you already introduced a little bit the company. So today I will be talking about AP321, a gene therapy treatment for FSHD. And before getting into this program, I just wanted to give a brief introduction to Epic Bio, who we are, what we do. So we're a platform company, and we're using a CRISPR platform, a CRISPR tool. We are founded by one of the CRISPR co-inventor named Stanley Chi, who really pioneered the use of CRISPR not to modify the genome sequence, not to cut the DNA, but instead to precisely control the expression of genes. And that's really at the core of everything that we're doing. And I think it will be explained through, the AP321 program that we'll go into more detail. We have a very diverse CRISPR toolbox to precisely control gene activity. And we actually have different programs, but again, I will illustrate that with AP321. In the context of AP321 and FSHD, we are planning to submit an IND by the end of the year, and we're planning to start our clinical trial early next year. We already have in vitro and in vivo efficacy data. I will show you a few slides on this. We have tested the efficacy of our product in a humanized FSHD mouse model that really confirms that our treatment is effective. And we also have done a number of safety studies in vitro and in animals that demonstrate that AP321 is safe. So as I mentioned, we are a platform company, so we also have other products utilizing the same platform. As you can see from this slide, AP321 is really the first and the most advanced product. As I mentioned, we're planning to initiate a clinical trial early next year. So how does our platform work? I know I mentioned the word CRISPR, and I might get a little bit technical in the next few slides. But I'm happy to answer question if this is not clear. So we're using the main component of any CRISPR system, which is a Cas protein. So that's the one you see here. It's the yellow blob on the slide. However, the major difference with other CRISPR company is that we're using a deactivated version of this Cas protein. So the Cas protein is really a molecular scissor that cuts the DNA, but we have removed the ability of this scissor to cut the DNA. So what it does is that it binds to the DNA and it stays there, but it doesn't cut. And that's why you see this little D in front of the Cas. It means deactivated Cas, so does not cut. Now, we're using a second element of any CRISPR platform, which is called the guide RNA. And essentially, the guide RNA is really the GPS of the whole system. It gives the coordinates to the Cas protein, and together they're able to go to the precise location of the genome we want to fix. And finally, the third element of the platform, which is really doing the magic in that case, is called modulator. So these modulators are attached to the dCas protein, and they are the one that precisely control the activity of genes through a variety of mechanisms. And I'll explain how that works specifically in the context of FSHD. So for FSHD, I think probably a lot of you know that the underlying abnormality is epigenetic in nature. What it means is that above the DNA there is a mark that is symbolized here by the circle. There is a mark that allows that specific sequence in the DNA to be silenced and not to be expressed. However, in FSHD individual, this mark called methylation is lost, and that leads to a de-repressed locus. And what it means is that the genes that are not supposed to be expressed are expressed in that specific locus. And the gene that we're talking about here is called DUX4. I'm sure most of you have heard about this gene. This is a toxic gene in muscle cells. And what it does eventually is that it causes muscle cell death. It induces toxicity and eventually kills the cells. So the product that we're developing, AP321, uses that CRISPR-based platform. You can recognize the yellow blob, the guide RNA, and finally the modulator. And so what we do is that we bring that product to the right location in the genome, and the modulator is able to fix the loss of methylation that is observed in FSHD patient. By doing so, we suppress DUX4 from being expressed, and then the rationale is that we stop cell death from occurring, and I will show you some data to confirm that. So another important element of the product is how do we deliver all of these components to the right tissue, to the muscle tissue in patients. Like it is the case often with gene therapy, we're using a delivery vector, and the delivery vector that we used is called an AAV. So that's how it looks like. And this AAV is really, it's administered via intravenous infusion, and it really what helps delivering the product to the right location. So AP321 is an AAV-based medicine. It will be administered one time via intravenous infusion to FSHD patients. The specific type of delivery vector, of AAV delivery vector that we're using for AP321 is called AAV-RH74. And the reason why we have chosen this one is because it has been tested in clinic already. And so we know that this product, this delivery vector is safe. We know that it delivers very well to the muscle, which is the tissue we want to target. There is a company called Sarepta that has used it successfully to develop one of their products that recently got approved. So, it really gives us confidence that the delivery vector that we're using for our medicine is safe. So I think that's an important point to mention. Again, I think I will get just a little bit technical here, but I wanted to show you what's inside this product, how it looks like specifically. So here you can see the AAV, that's what it looks like. That's the delivery vehicle that contains the different elements of AP321. And if we look a little bit closer, that's how it looks like. So essentially you can see here that we have the GPS that I mentioned, the guide RNA that target that specific location in the genome. Then we have our Cas protein that we called DCAS onyx. And then we have the modulator that act through methylation, and that is fixed to the Cas protein. As an additional layer, we have here what we call the promoter, a muscle-specific promoter. This is really to make sure that our Cas protein and modulator is only expressed in the target tissue type, so is only expressed in the muscle. So I know this is a little bit technical, and there might be some things that you may have not understood from that slide, but what I really wanted to highlight here, the main takeaway is that, number one, this product is safe when compared to other CRISPR editing technology. As you can see, again, we do not induce any DNA cuts. We do not induce any change to the genome sequence with our technology. Number two, our product is precise. You've seen it with that, GPS, but also by the modification that we induce. We're only targeting that specific site in the genome, and we're only changing methylation. Finally, delivery, as you can see, by delivering this product with an AAV, and because of the way AP321 works, it only requires one intravenous injection. It doesn't really require any repeat administration of the product. So that's a one-time product, one-time delivery. So just to show you a little bit of data that we have generated. So here, this is data that we have generated on patient cells in the lab. We have gotten patient cells, muscle patient cells from FSHD patients, and we have tested our product directly on these cells on actually different patients. And we have summarized all of the data here. So on the left, you can see that when we treat FSHD cells with our product, we observed a decrease of the entire DUX4 pathway. So not only DUX4, but all of the downstream genes that are known to be activated with DUX4 are suppressed upon treatment. And that is validated not only in one cell line, but in multiple patient cell lines with different background, different size of the D4Z4 repeat, different severity of the disease, patient with different age of onset. So that really shows that the product suppresses the cascade in a variety of contexts. On the right, similarly, we've tested that on multiple FSHD patient muscle cells. As I mentioned, we know that DUX4 induces cell death. That's something that has been documented in patients and also in vitro, or also in patient cells in the lab. This abnormally high cell death is well documented. However, here again, when we treat the cells with AP321, we're able to rescue cell death. So we're able to reduce cell death and rescue cell survival in this context, which is really the main phenotype that affect FSHD patients. And so that really confirms that the product works not only at the molecular level, but also rescue cell survival. We have tested exactly the same thing in an animal model. We have used a humanized FSHD mouse model. And you can see on the left that in mice that are humanized with normal human muscle cells, we cannot detect DUX4 as it is expected. There's no DUX4 in normal human muscle cells. However, in mice that are humanized with FSHD human muscle cells, we can detect DUX4 as it is anticipated. Upon treatment, we're able to reduce DUX4 from being expressed. Here, it's only one piece of data, but this is confirmed with, as I mentioned, the entire DUX4 downstream cascade. On the right, you have the same phenotype looking at cell death. The first bar here represent what's happening with normal human cells. They survive well in this model and repopulate. However, when we use FSHD human muscle cells, you can see that they don't survive as well, and there is this drop compared to normal. Upon treatment, we're able to rescue cell survival in these animals of 55% compared to the non-treated control. So these data essentially illustrate the same finding that what we found directly on patient cells. Here, we're demonstrating this in animal model. And again, I just wanted to show a little bit of data in cells and in vivo. We have presented more comprehensive data set at multiple medical meetings, and I'm also happy to give you more details for each of these slides. So as I mentioned, we have also tested the safety profile of AP321. So here I summarized what we have done into 4 categories. So number 1, we have looked at the potential immune response on human cells, on human immune cells directly. And the reason for doing that is we know that using CRISPR technology and other Cas protein that are very different from the Cas protein that we're using, we know that sometimes there is an immune response in human. We did not anticipate the same problem with our technology because we're using a very different protein, the Cas protein. The Cas protein that is used by others is derived from bacteria, so may trigger an immune response. This is not our case. However, we wanted to measure that. And so we've taken human immune cells and we've tested their response to our Cas protein. And we have found that none of the cells tested responded to our Cas protein. They did not develop they did not show any immune response there. That really helps understanding the safety profile of the molecule. Of course, we tested the product in animals, not only in human cells. So we've done a safety study in mice. We have found the product to be safe in that study. Similarly, we have done a study in monkey, and we also have found the product to be safe in that study. And now we're in the process of doing what we call GLP study. So it means that it has a level of regulation that is compliant with what we need to do for IND submission. Those studies are ongoing, and that's the last piece of the safety study before we can submit an IND. So far, all of the data that we have fully analyzed from these studies indicate that AP321 is safe. And there are 3 particular points that I wanted to highlight. Number 1, and I mentioned that before, but the delivery system AAV-RH74 has been safely tested in clinic already and is part of an approved product. So here we already know that this delivery system is safe. AP321 does not have the same safety concern as other gene editing technology. Specifically, we do not modify the genome. We do not cut the DNA. And finally, from what I have shown you here, AP321 has demonstrated safety in animal model and in human cells from the data that we have analyzed on these safety studies. So moving on to what we're planning to do in clinic. So early next year, we're hoping to start a clinical trial for AP321. It will be the first in human. It will be an open label trial, a dose escalation study that will mainly focus on the safety and tolerability of the product. It will be administered one time, as I mentioned, in patients from age 18 to 75 years old. It will be a small study between 6 to 9 FSHD patients. The study will last for 12 months. And like it is the case for any gene therapy, we will have a 4-year follow-up. We're planning to have several clinical study centers in the US, potentially Canada, the UK, Germany, and the Netherlands. So one thing that may be interesting to a lot of you is what are the inclusion and exclusion criteria for the study. So some of the key inclusion criteria are listed here. We will enroll patients that have a confirmed genetic diagnosis of FSHD type 1. Patient need to be able to give an informed consent. As I mentioned, patient will be between 18 to 75 years old. We will also enroll patients that range in the intermediate category of a Ricci score, which is a clinical severity score. So I think some of you might be familiar with this Ricci score, and we'll enroll patients that are between 2 to 4. As part of what we will measure, even if the study is mostly focused on safety, we're going to have a lot of measure to evaluate the efficacy of the product. Part of this measure includes MRI, so we want patients to have no issues with MRI. As it is the case with a gene therapy trial, we want patients to agree to use contraception for the duration of the study. And we will enroll patients with normal liver and kidney functions. Some of the key exclusion criteria are listed here. Since we have a number of functional tests that requires the ability of patients to walk, to perform the 10-meter walk-run test, we will exclude patients that are unable to perform the 10-meter walk-run test or that use a walker or a wheelchair. We will also exclude patients that have antibodies against the delivery vehicle that I mentioned, AAV-RH74, or against our Cas protein. Even if we don't anticipate that this will be the case, we will measure that in patients, and we will exclude any patient that shows these antibodies. We will also exclude patients that have significant medical issue or allergies to the product that may prevent them to be enrolled and participate to this trial. Patients should not use drugs or supplements that affect the muscle function. They should not be enrolled in another FSHD clinical trial. They should not be pregnant or breastfeeding. And we will also enroll patients that are less or equal to 90 kilograms in weight. So I think this is also potentially very interesting and might trigger a few questions. So first of all, I'd like to start these slides by saying that we do have a patient survey ongoing to make sure that what we're proposing here is aligned with FSHD patients, and we can talk more about this at the end. But this is to give you like a very high-level summary of this clinical trial and what to expect from it. As I mentioned, it's a 1-year study. We will start screening patients 30 days before treating them with EP321. And at the time of screening, they will be, patient will go through different things. So first of all, we will go through informed consent. There will be a number of safety tests, physical exam, blood and urine tests, also echocardiogram and electrocardiogram will be taken at screening. Finally, there will be efficacy tests, so that is really to measure at baseline, the level of the disease severity before we start the treatment, and so that will include MRI, as I mentioned, also a number of functional tests, the clinical severity score, a number of questionnaires, and a needle muscle biopsy will be taken. 3 days before the treatment, we will start a course of steroids in patients. And then eventually at day 0, patient will receive EP321 treatment. For the first month, we will follow up with a lot of safety tests to make sure that everything is okay post-treatment. They will essentially be very similar to the tests that were performed at screening. And starting at 3 months, we will look at efficacy. So we will repeat all of the tests that have been done at the time of screening, and we will also take another muscle biopsy. At 6 months, it will be the exact same series of tests, except that we will not take a muscle biopsy. And finally, for the last visit at 12 months, we will have the same safety assessment and efficacy test, including one last muscle biopsy. So I wanted to illustrate, what might be AP321 clinical journey. So we talked about what you're planning to do for the first in humans. So this is really just that first phase of the clinical trial, focused on a small cohort and focused on safety and dose finding via an efficacy signal. After this first phase, we will move to another phase of the clinical trial. At that stage, we most likely will have a final dose selected, and that phase will be focused on safety, but also getting really compelling efficacy data to move to the next phase. Finally, the third phase of the study will be focused on safety again and efficacy on a larger cohort. If everything works well, with these 3 phases of the clinical trial, we might be able to get approval for AP321. Of course, this is a longer term, but we wanted to illustrate what the clinical journey might look like for the product. All right, I will stop the presentation here. Thank you, everybody, for your attention. And Casey and I are happy to answer any question that you may have. Thank you very much.
Thank you so much.
Thank you. Before we get started with the Q&A, I would like to just mention how rapid your drug development process has been. During your presentation, I took a look, I think, at the first meeting we had, which was almost 2 years ago. I think it was in July of 2021 when you first reached out and you mentioned your interest in FSHD and some very preliminary studies you had done with some cell lines that were available through Coriell. And I have to applaud you and the team at Epic about how quickly you have developed this program and how quickly it's advanced, especially with the animal studies and the more complex delivery studies, because I think the Sarepta virus was also kind of being developed at that time, and you guys made some really good decisions very early on to be able to move this program to where it is now and to kind of be on the cusp of clinical trials, essentially. This is really amazing.
Thank you very much for saying that, and I will quote our CEO on this, but I think it's really a testament to the team we have here at Epic, and we have people around the table that have experience already in drug development, so that really helps making some of these decisions early on. And another element that I think is very important, and the reason why everybody is excited about CRISPR and talks about CRISPR is that it really facilitates, because of the programmability of it, it really facilitates developing a medicine more quickly than with some other platform. And so I think this combination of a very robust platform and having a team that were able to take the right decision at the right time explain why we were able to move that fast. But thank you for saying that. Appreciate it.
Oh, my pleasure. June, would you like to start with question, or should I you're on mute.
I will try and see how my voice holds up. So we have a slew of questions relating to the mechanism of AP321. Could you expand on the methylation mechanism initiated by the modulator, if that's not proprietary information? And somewhat related to that, so, other people are asking if you have looked for, I suppose, off-target effects, so hypermethylation in other regions of the genome.
The length of, there were some more questions related to that about how long you expect the methylation to last. Is this a lifetime? And as muscle renews and maybe new muscle gets added, would that be protected? Is the therapy designed to slow down the disease, stop the disease, and will help regenerate new muscle? So this is a lot of questions that a lot of people are curious about.
So I think there's three main questions, so I'll try to answer them all, but if I forget one, please remind me. So the first question on the modulator and how exactly it works. Essentially the modulator are part of protein, and what we're doing here is that we're essentially mimicking what the cell is supposed to do by itself. So we're using similar mechanism that is already present in the cells that are known to add a methylation mark. And so we're using this protein as part of this modulator. And so they are the one that when in contact with the DNA, they will add that methylation mark to the DNA. I should say, and I think it will partially answer the second or third question. We're not the first one to discover this type of mechanism. Thankfully, there are decades of research on this type of epigenetic mechanism. We really benefit by the years of research that has been done in this context. And through that, research that has been done, and also through research that we have done in our own company, we know that when we impose a methylation mark that is stable through time, through cell division, that is mitotically stable. So what it means is that, and people have studied that, it stays for a very long time. I think as long as people have studied it, it stays for years. Of course, specifically with our product, we're not able to say whether or not it will last for a lifetime, because we would need to have done those studies for a lifetime. But we have done studies where we look at, in vitro after multiple cell division, and we still observe, suppression of the gene of interest or this type of thing. So we know it is long lasting. And to corroborate that, we know the mechanism of action. We know that when we add a methylation mark, it stays also through cell division. But we're also delivering our technology with that AAV delivery vector. And we know that AAV stays in the cells for a very long time. And there is preclinical data, clinical data now, because you're not the first one to use AAV for gene therapy. And in the muscle cells, or in the muscle tissue, we know it stays there for decades. We have these two levels of persistence of our product via the delivery vehicle that we use and via the mechanism of action of the methylation that gives us a very strong confidence that it will remain for a very, very long time, if not the life of the patient. That's not a question that we could answer at this point, but for a long time, that's for sure. So there was a question on the mechanism, a question on how long it lasts, and the third question was what is anticipated if it will stop disease progression or if it will improve. So I think, and Casey, please feel free to answer this one. So we don't know until we go to clinic, to be completely honest, but we have discussed this, and that's always a question in gene therapy. Is it going to stop or is it going to improve? And at the very least, we believe that it will stop disease progression, but we have good rationale to believe that it may improve the disease trajectory. And I think Casey can also answer that. But what I would like to point out is that the cells in FSHD patients, the muscle cells are healthy. Until they get poisoned by DUX4 and eventually degrade and die. But there's no inherent problem of contractility of these muscle cells, like it is the case for other muscular dystrophy. These cells, if I compare with Duchenne, for instance, in Duchenne, there's a protein that is critical for the cells in the tissue to contract that doesn't function properly. It is not the case with FSHD. So if we can really stop the cells from dying, if we can rescue cell survival, we're in a very different place than for other muscular dystrophy. And there is a good rationale that we could improve, the disease progression here. But Casey, if you want to add anything to this, please feel free.
Sure. So thank you for the opportunity, and thanks to all of you from the various parts of the United States and across Europe and other places for joining this. First of all, I have to say that as a clinician scientist, this is an amazing technology that has developed incredibly quickly. I actually never thought in my career I would see a potential treatment for FSHD that actually gets at the root cause of the disease. And that's exactly what EPI-321 does. It gets at the root cause of the disease by sort of normalizing the methylation of that sequence. So this is a very interesting and important question about muscle. So as a skeletal muscle physiologist, I can tell you that skeletal muscle is very plastic. In other words, it has the ability to regenerate itself. In fact, in mice, you can completely ablate an entire muscle, and as long as the surrounding fascia and structure of that muscle is intact, the muscle will completely regenerate within 21 days. So muscle is plastic. It does have the ability to regenerate, with the exception of many of these muscular dystrophies. So FSHD is a muscular dystrophy. However, Alex is absolutely correct. Really, there are no contractile protein problems in the muscle cell. The muscle can contract normally. What happens is that it's being poisoned. The cells are being poisoned by DUX4, and therefore many of the cells will die over time. If you can stop that cell death, then what you have left are really normally contracting healthy muscle cells. So given the sense that muscle is plastic, it does have the ability to regenerate. My personal hope, and we'll have to see in the clinic, my personal hope is that the muscles will stop degenerating from lack of expression of this poison, this DUX4 poison, and then be able to contract normally and behave normally. So things that individuals do to improve the health of their muscle, exercise, diet, just normal activities of daily living. My hope is that strength and mobility will actually improve, but that's my personal hope. But we'll have to wait and see. So that's my own perspective on this, and it's a very important question, both from a scientific and medical perspective. So thanks for that opportunity to share, Alex.
Yeah, thank you very much. I think this is one of those difficult things to predict because as muscle atrophies and sometimes there's fatty infiltration, fibrosis, those scar tissue essentially might prevent muscle to regenerate normally, but the body is amazing at clearing up some of those things. So we don't know long term really if there, and our hope is that muscle is plastic enough that the body will be able to clear out some of this debris and maybe allow muscle to regenerate. And I don't know if hopefully there will be indications from other diseases that may be a little bit ahead of FSHD to kind of give us a preview of what to expect. There were some questions. I don't know if this was covered. I'm sorry. Every time I ask questions, the leaf blowers tend to turn on just outside my window, so I apologize for that. But are there any predicted side effects? And since this is a gene delivery, what would happen if adverse effects are discovered? Is there a way of switching it off?
Yeah, Casey, do you want to answer this question? I'm happy to do so as well, but I thought it might be a question for you.
Yeah, so we actually anticipated this question, and let me just reiterate some of the things that Alex said. First of all, the delivery vehicle, if you would, the AAV, is actually not only been proven safe, it's actually approved recently in the US for Sarepta's product. So that's comforting to know that. But I just want to kind of make some general statements about safety and potential risks here. With any clinical trial, particularly any first-in-human trial, there are inherent risks. And the most important thing is that you, as a potential patient in a clinical trial, be well informed. So the most important thing is to speak to your personal physician and to carefully consider all of the potential risks and benefit to yourself as a person. So in a clinical trial, there is something called a consent form. And in the consent form, there will be very detailed descriptors of potential risks and potential benefits of the treatment. Because there are ongoing studies right now that EPIC is continuing to work on, those are not completed at this time. So it would be better to be best informed, accurately informed by your physician. So I know I didn't address the question directly, but please be patient and carefully consider the risks of risks and potential benefits of this treatment with your physician. That's probably the best advice I can give those listening.
And I think that thank you, Alex, for including that slide explaining the different phases, clinical trial phases. The FDA obviously is very concerned about safety, and so that's how the whole study is, the pipeline is set up, that there needs to be a lot of safety data presented before they allow an IND into first-in-human trials. So all of those tests have to be done in different animal models for different durations, and these are all the work that you guys are currently doing. And then the clinical studies start off small with dose ranging studies, just to kind of address and kind of provide some information on that. And of course, all of the decisions, all the calculations are made to always be in that safety area. So thank you. That's, I think, a very a very logical approach.
Yeah, thank you for saying that as well. This is very true. We'll have the full picture in the informed consent, for every patient once we get there. But indeed, we're running those safety studies, and as part of what we will submit to the FDA will actually evaluate the benefit risk. And if the study is up, and it's because the benefit risk ratio is in the favor of the benefit. And as I mentioned, from the safety studies, that's what we know of the product. Studies are still ongoing, but as Casey pointed out, this AAV has made it to clinic already successfully, safely tested. There are always potential risk with clinical trial or gene therapy trial, but there's important information about what we use in this product that I think is relevant to the safety.
There was a comment actually just came up. If you could stop your screen sharing, I think people wanted to be able to see you more closely. Thank you. And June, you're on mute.
Sorry about that. Yes, I was just about to ask about the screen sharing, but thank you. Well, this is related to the preclinical, testing of the safety, I guess. And the question's wondering so AAV has a history of having some liver and possibly cardiac toxicity. With this construct, what does that look like right now?
Yes, so again, I just want to preface that some of those studies are still ongoing. But I can talk about what we have fully analyzed at this point, and we have monitored liver function, and we have also monitored cardiac function. So first of all, that's true that AAV has a history of a little bit of liver toxicity, cardiac toxicity. I think it is clear from what has been tested preclinically and clinically that not all AAV are equal on that particular aspect. And again, the AAV-RH74 didn't have the same issues than some other AAV have faced. So I think it's an important point just right there. From the preclinical studies, so I mentioned we've done serum chemistry, looking at liver markers. In the NHP study that we've completed, we haven't found, any red flag. Looking at the serum chemistry, also looking at the histopathology, so looking at the liver in detail. There's no major red flag here that was reported from this study. We have also looked at troponin, which is a marker for the heart. We've done EKG and another way of measuring the heart function. And again, we haven't found any abnormality from these studies that we've completed on monkeys. And same story on mice. So we need to have the complete picture. But I think we will be able to identify a dose that, like those studies report, that is safe for the first in human, and also knowing what happened with AAV-RH74 in human as well, that there hasn't been major red flags compared to other AAV clinical trial.
So here's an interesting question. DUX4 expresses in male testes with unknown function and in 4-cell stage of early embryonic development as one of the key zygotic genome activator drivers. I wonder if the patients could generate normal germ cells and would fertilized eggs survive the zygotic activator stage after EPI-321 treatment?
Yeah, I think that's a very good question, and that's why in that slide I mentioned the additional control we have. We have what we call a muscle-specific promoter. So as an additional layer of safety, we made sure that the product is, preferentially expressed in the muscle tissue. We don't believe that there is expression in the cells that is, required for development in that sense. That being said, it's part of the safety studies that we're doing. We're looking at what is going on in the testis, and so we can make sure that we don't have any expression in cells where DUX4 might be needed. That's another advantage of AAV gene therapy. You can control where your product is expressed. And that's not the case with other technologies, but here it gives us an additional layer of safety to make sure that we don't affect DUX4 expression in other tissues.
There's some questions about what will you be measuring in muscle biopsies in the clinical trial, and what do you hope to see with the MRI? And finally, are you partnering with researchers who are doing natural history studies? So are you, I guess maybe the underlying question there is, do you feel like you have the clinical endpoints that you need for this trial?
Yeah, so maybe I can answer the biopsy question, and Casey, you can answer the two other questions. We're hoping to get a lot of information out of the biopsy. We will, number one, look at the methylation level. We want to make sure that the product does what it should do, which is remethylating that region in the genome. We also want to look at DUX4 and the entire DUX4 cascade of expression. We also want to look at potentially, if possible, we want to look at the level of cell death and if we manage to stop that. Harvesting from a needle biopsy, we're hoping to generate that information, at the molecular level, and also if we can address the phenotype of cell death, if we can stop cell death. Casey, feel free to add anything to the biopsy question, but I think you can answer the two others.
Sure. No, I concur with your answer to the biopsy question. The other two questions, I'll answer the third one first, and that's yes, EPIC has been very fortunate to have access to a very large human database of natural history in FSHD, both in the US and abroad. So we've had the really great fortune to be able to analyze that data and to help inform the clinical endpoints. I think the trial is designed not based on hypothetical stuff, but it's actually based on real data in real patients. So that's a huge, just a very unusual and very fortunate opportunity here to design the EPI-321 first in human trial. So in terms of MRI, that's one of the endpoints, what we call an exploratory endpoint, that was not included in this very large database. Now there are some published data on use of MRI, whole body MRI, to look at things like the volume of muscle, how much water content it contains, so forth and so on. So we're looking at this as an exploratory endpoint. Whether or not it would be that useful in terms of efficacy, we don't know. But I think it would add to the fund of knowledge of the disease. And perhaps it could give us some insight into more of the macroscopic view of the muscles throughout the body. With a muscle biopsy, you're taking a very, very small sample from one muscle, whereas a whole body MRI can give you a picture of how large the muscle is, what the level of inflammation is in the muscle, and so forth. And so this would be an exploratory measure. And again, we'll just have to see what the data look like to really determine how much useful information is gained from that. But it certainly will add to the fund of knowledge of FSHD and hopefully provide us some further insight into how the entire musculoskeletal system is responding to this treatment.
Great. There's a question about whether this treatment could be used for FSHD too.
Yes, and I can answer this question. I think for sure we've done most of our work preclinically on cells that are derived from patients that are type 1, FSHD type 1. Now, if you consider the mechanism of action of our product, we believe that it will work similarly on type 2, FSHD type 2, but we would need to test that before we can say that for sure, which is why for now we're starting with FSHD type 1. But, since we're not changing the DNA sequence, we're just adding a methylation mark, and that's also the underlying problem in FSHD type 2, so it should work similarly in FSHD type 2.
Just adding to that, theoretically, in FSHD type 2, you actually have more D4, Z4 repeats that could be methylated. So one might even, predict that maybe the drug might work more effectively in FSHD type 2. This is obviously, until you confirm it experimentally would be interesting hypothesis to test.
Yeah, no, that's a very good point. I think, yes, until we test it, we would not know, but you know, we've had similar internal discussion. Very good point.
There are only a few minutes left, but there's a lot of questions. I think people are very, the audience is very excited about this, and there are you know, a lot of questions on when is the next phase is going to be, and when can we expect FDA approval, and will the criteria from phase to phase change over time? Do you anticipate that happening?
Casey, yeah.
Yeah. So, you know, there is a sort of an overall plan on the various phases, but honestly, it's too early to really even speculate on the timing. I can tell you that the people at Epic Bio are absolutely dedicated to moving this process forward as fast as possible, and to get an approved treatment for FSHD that really addresses the root cause of the disease. So we'll see. I have high hopes that things will go well. I don't think that we're going to have an issue with recruiting patients. You know, we're very fortunate to have this very large FSHD natural history database to really inform and create a very strong clinical trial. But you know, each phase of the trial will inform the next phase, and so you know, the idea here is that with each iteration of a larger trial, you get you better control over your efficacy endpoints, your assays, and so forth and so on. So there will be improvements over time, and the process will go as fast as possible. That my reassurance to those listening is that this will go as fast as the company can accelerate the process.
Thank you.
A couple of other questions that were asked a number of times. One is if you've previously been in a trial or on treatment for something like AOC 1020, so the mRNA therapies, does that kind of therapy preclude you from having this therapy in the future, or is there a washout period after which you could take it?
Yeah, well, I can answer maybe the first part of that. If you are involved in another clinical trial for FSHD, it would preclude you enrolling in EPI-321. Now, that's not to say that if you're, you know, on or have taken a medicine such as a corticosteroid, certainly there can be a washout period, and that should not preclude. But that is one of the general exclusion criteria of any clinical human trial. Is that you can't be enrolled in two trials at the same time because of obvious scientific issues there.
I think people understand, but I think there's a possibility over the next few years, people start to be on different treatments. And then
Yes.
If this new treatment comes along, can they stop that other treatment and still be, you know, so.
Yeah, I think it'd be probably a case-by-case basis, but most likely, yeah, I could see that as a potential. And maybe Alex has some thoughts on that as well.
Another question we get a lot of, I'm sorry, because we're almost out of time, is, but I did want to slip this in, is what about children under 18? Wouldn't that be the population that would be most benefit from this? And what are your thoughts about?
Sure. No, that's a very good question. Absolutely, because younger patients often have a more severe disease. There is actually a legal reason called the Code of Federal Regulations, published by the FDA, that says that, you know, if you have available, if there are adult patients, that those there needs to be evidence of patient safety in adults before you can age de-escalate into children. So we're actually prohibited from doing that immediately in patients under the age of 18.
Very good. Okay, well, we are sadly out of time. There's still many questions. Maybe we can send you some of those questions offline, and if you'd care to answer them, we can post them on our website. And I do want to mention we will be in the next day or two sending out an email with a link to the survey that Epic Bio would like patients to participate in to find out about their preferences in terms of participating in clinical trial. So Beth, over to you.
Yes, real quick too, I just have one quick question. When do you expect the trial to be listed on clinicaltrials.gov?
Our hope is that the trial will begin in the first half of next year. So sometime in that time period, I would think it would be listed on clinical trials. Yeah.
There were a lot of questions about the timing, and I don't see it on clinical trials yet. So
right, you won't.
Our recommendation, just keep checking between now and January of 2024. Okay.
Yeah, and please
Great.
answer the summary of the questionnaire that's coming out. That would really help us inform better on the clinical trial design. So we appreciate everybody that can answer those questions.
Excellent. All right, you heard that, everybody. Answer the survey that's coming out. I just want to thank everyone for attending our FSHD University webinar today. A very special thank you to Drs. Collin and Childers at Epic Bio for all of the exciting updates and information, and for answering our plethora of questions today. And thanks to June and Jamshid for coordinating those for us. I appreciate that very much. It's interesting you mentioned that, Dr. Childers, because our next FSHD webinar is on August the 24th. It's entitled FSHD Experiences and Insights from Europe. Our speaker is Megan McNiff. She's a research associate from the John Walton Muscular Dystrophy Research Center at Newcastle University in the UK. But she's presenting the results from the FSHD Europe patient survey. This study was actually aimed to understand the FSHD community's perspective, so that when clinical trials are developed, they're designed and organized in a way to actually maximize patient involvement, which is a growing interest from the pharma companies, right? Other than that, just be sure to visit our website events calendar for all of the upcoming chapter, wellness, and educational events. Thank you again, doctors, for joining us today, and we'll see everybody next time. Much appreciated. Take care, everybody.